Welcome to the Fungisphere webinar series, brought to you by the Sydney Infectious Disease Institute here at the University of Sydney. I'm Associate Professor Justin Beardsley, and it's my pleasure to introduce these talks, our passionate team, and share some of our mission with you. We're dedicated to increasing our understanding of all things fungal, be that human health, animal health, agriculture, ecology, or even the productive and industrial use of fungi. We are seeking to uncover the threats and the promises offered by these remarkable organisms. This webinar series is more than just a series of talks. It's a platform to bring experts in the field together to share their knowledge and hopefully to build new connections. It's a testament to the synergies that exist between researchers here, but also colleagues globally. Through these talks, we share some of the incredible work being done here in the University of Sydney, but also provide a platform for international experts to share their groundbreaking research too. So, whether you're a seasoned researcher, a student, or simply somebody with an interest in the fascinating world of fungi, you'll find something of interest in this webinar series. Together, we're going to explore the threats and the promises of the fungal world. In this popular talk, Dr. Johanna Rhodes from Imperial College London presents some of her work on antifungal resistance. What's driving antifungal resistance in the environment, and can we prove that this is leading to drug-resistant infections in people? Let's hear from Jo to find out.
Thank you. Thanks for having me. I know it's a late one for you guys and an early one for me-ish. So yes, I'm going to be talking about acquisition of drug-resistant fungal infections from the environment. I'm going to be focusing primarily on Aspergillus, touching briefly on Auris, given sort of like some of the recent stuff. So yeah, I started at Imperial 9 years ago. This year is my 9th year with Matt at the helm, and just basically moved from crypto to Aspergillus to Candida, and then just kind of like working on all three. And then I'm just basically now working on, if there's a fungal outbreak, I work on it. So I just basically work on anything. I primarily do just bioinformatics and genome sequencing analysis now, and I leave the lab work to much more talented people than me. So I'm just going to start my talk. I know my desktop looks incredibly busy. Does that all look okay for everyone?
That's perfect.
Cool. Okay. So we recently renamed ourselves to the MRC Centre for Global Infectious Disease Analysis. Previously, we were just Department of Infectious Disease Analysis. So we've got a snazzy new title. So obviously, you all know that fungi are ubiquitous. They diverged from the animal and plant kingdoms approximately 1.5 million years ago. And there's thought to be about 150,000 fungal species, but it's estimated to be higher. So there was a paper that came out a couple of years ago that said that there could be anything between 2.2 and 3.8 million species. So our knowledge of fungi in the wild is pretty limited. About 8,000 of these species cause disease in plants. Obviously, we've seen that recently with things like ash dieback here in the UK, and then obviously elsewhere around the world, we've seen fungal infections of banana plants, chocolate plants, cocoa plants, which is devastating, coffee, or the works. And just over 600 associated with humans as commensals or pathogens. So actually it's a really small amount that have kind of spilled over into plant and human diseases. So when I first joined the Fisher group, he just got this really nice paper out in Nature to show that fungal outbreaks over recent decades are increasing. And obviously Matt's work is primarily focused on Batrachochytrium and the mass amphibian diebacks. And then we've started to sort of branch out into other wildlife diseases, so white nose syndrome. And recently we've been looking at aspergillosis in the kakapo parrot over in New Zealand. So we've been part of the kakapo aspergillosis consortium as well, looking at that. You've got ash dieback and other plant fungal infections, and then of course you've got the human infections like cryptococcus. So the total number of alerts of both animal and plant fungal infections are increasing. And basically we want to figure out what's driving that. So again, Matt sort of collaborates with Sarah Gurr quite a lot and just came up with this nice little schematic of how environmental change is driving new patterns of fungal disease worldwide. So basically, we know that we can get selection for virulence and resistance to antifungal chemicals artificially, so artificial selection, just basically through the way that we farm, basically through, you know, how we grow crops. We have a monoculture, genetic uniformity over large areas. We are using similar drugs to similar class drugs to what we use medically as well. But also there's these anthropogenic opportunities, so climate change. So Arturo Casadevall is quite hot on this at the moment. It's just like how climate change is changing sort of species distributions and perhaps has contributed to the emergence of pathogens such as Candida auris. And we're also contributing to long distance dispersal. So we're moving, well, perhaps not in the last year, obviously, but we are moving ourselves. You know, we're going on exotic holidays, we're going to like all these fancy conferences and all this kind of stuff. But we're also moving stuff around the world. We're moving fruit, veg, meat, and also pets as well. So like the amphibian mass dieback, we think originated in the Far East, and basically through the amphibian pet trade has spread across the world. So we're moving new pathogens to new places just by our activity. So every single year, not this year or last year, unfortunately, Matt goes to the Pyrenees and goes to the same lakes to basically count the number of amphibians that are visibly dead from BD, and also to take samples to just kind of you know, test and make sure that it is BD and which lineage of BD. So this is a picture of him here, kind of like trekking through the mountains and coming across this fungal sac and just sort of noticing how the spores are just easily dispersed on the air. And as humans, we're exposed to environmental fungi every single day, and basically depending on the size of the spores, they can sit in different parts of the lung. So if they're slightly bigger, they sit a bit higher. If they're slightly smaller, they can get down into the alveoli. And you know, we're really interested in all of these, and we're adding to them all the time. So so we're seeing some outbreaks of some pretty weird pathogens at the moment in our COVID wards, in our COVID patients. So a few years ago, pre-COVID, we decided to set up this Burkhard 7-day spore trap on the roof of St Mary's, which is the hospital in Paddington, which is where I work. And we also set one up in a rural location in Hertfordshire, which is sort of like one of the home counties. And I'm not in this photo because I have a fear of heights, and this wall literally comes up to like your thigh. And I was just like, I'm not doing that, not a chance. So, you know, we these three lovely people, Rebecca, who's the health and safety person, Jen, who's a PhD student. I'll talk a little bit more about her work later on. And this guy's called Slim because, you know, he's fairly slim, and he's the maintenance guy, and he was the one doing all the heavy lifting. And basically, you know, you just change the tape every single week, and it just filters 10 liters of air per minute. And basically whatever's in the air just gets stuck to the tape, and then you can just chop it up and PCR it, or, you know, or sequence, Sanger sequence, whatever you want to do to see what's present. So we were expecting to see more fungi in the rural location than the urban location, just due to the fact that we were using azole antifungals in sort of like the rural locations due to crops and things like that, and see less in urban because of pollution and things like that. And we found loads of different species attached to the tapes, and I'll be talking predominantly about Aspergillus. But what about resistance? So I'm sure you all know, but resistance occurs in all classes of drugs used against plant and animal fungal infections. And the 6 main classes of fungicides are the morpholines, the azoles, you've got the MBCs, the QOIs, and then a couple of others which I can't remember. And basically what happens is that you've got dual use of these azoles, both in agricultural settings, but also in animal settings and human settings. It's a little bit like the antibiotic problem in that, you know, we use antibiotics agriculturally to make chickens bigger, to put on weight. You know, we use them in our cattle, but then we also use them medically as well. And what we can see is that the resistance to azoles has increased throughout time in both animal and human diseases. And so we are really interested in seeing and in testing this hypothesis that human fungal infections that are drug resistant are originating in the environment. And apparently this is a controversial topic. So this is just an idea of the amount of azole antifungal drugs that we use in the UK in agriculture. So since 2000, there's been a 6-fold increase in the application of azoles, and the UK sprays about 250 kilos of azoles per year as crop protection. And this graph here, it just gives you this census ward level estimates of the usage of fungicides just for England for the year 2000. You can see that there's some dark areas where there's lots being used, and then the lighter areas is where there's hardly any being used. And you would, you know, that's what you'd expect. You'd expect sort of like London to not have any, and this is Birmingham here, so you know you expect those places to not have any because they're just highly urban. Not quite sure why the Isle of Wight doesn't have any, because I know that there's quite a few farms there, but it's also due to, you know, reporting factors as well. But if you compare this to what's used in the Netherlands and Denmark as well, the UK as a whole is actually using more than our European neighbours. So the predominant aerosolized fungi that we were spotting on the Burkhardt in the Paddington spore trap was Aspergillus. So for those of you who aren't familiar with Aspergillus, it's a fascinating organism. It can grow up to 60 degrees C in soil and compost, and it sporulates profusely. So this is a little figure that I've swiped from one of Paul Vervet's papers, where he talks about the 3 sort of life cycles of Aspergillus. And it's got a sexual cycle where it creates these cleistothecia, and then you've got the asexual one, which is thought to be the most predominant, but then it's also hypothesized that there's a parasexual cycle as well. So you've got these really complex life cycles that may or may not exist, we're not sure. We think they do, and basically there's opportunities here for recombination to occur through the sexual life cycle, and so it's like, you know, the generation of new combinations of resistance polymorphisms, and then when it reproduces asexually, it just has this capacity to just have a widespread dispersal of something that is potentially highly fit. So yes, this is a compost heap. I'm going to talk a little bit about Jen's work. So Jen's PhD has predominantly been on environmental aspergillosis. So she was just literally going around the country picking up soil samples from compost heaps and just noticing the amount of aspergillus that grows and the amount of drug-resistant aspergillus that grows. So I'll talk a little bit more about her work in a minute. And the patient cohorts that are at risk of aspergillosis, the main one is cystic fibrosis for us. So the UK has the 2nd largest cohort of cystic fibrosis patients in the world, the first being the US, and London is kind of like the main centre. And Imperial NHS seems to have the largest cohort in London, because we do a lot with respiratory medicine. And we've also got, to a lesser extent, neutropenic transplant patients, a few asthmatic. Very rarely do we see it in HIV/AIDS, cancer. And then just recently we're starting to see influenza-associated aspergillosis. And then obviously over the past 12 months, we've started to see COVID-associated pulmonary aspergillosis as well, which is rather concerning. And there's been reports from mainland Europe that the mortality rates can be quite high for people with Kappa. And the link with these, we think, is the use of azoles in agriculture and also medically. So for those of you who aren't aware, azoles target the Aspergillus ergosterol cell membrane biosynthesis by inhibiting the CYP51 enzyme. They do this in two ways. So firstly, they do an overexpression of CYP51. So there's duplications in the promoter sequence which increase the expression of CYP51A. So the predominant ones are TR34, so a repeat of 34 base pairs, and TR46. We're also seeing TR53, TR130. We're seeing all combinations. Basically, you come up with a number, we've probably seen it. And then there's also the structural polymorphisms, which will interfere with the entry of the azole molecules into the enzyme's hydrophobic access pocket. So I did a paper with my ex-husband a few years ago, which basically looked at the L98H, which is this one here, which is predominantly seen with the TR34 tandem repeat. And the L90H on its own is capable of making the volume of the pocket smaller, so that the azole molecules can't get in, and it's just inhibiting the action there. And there'll be similar sort of actions for all of these. And again, we're seeing combinations, different combinations, new polymorphisms coming up all the time. So Ali Reza Abdul Rasuli was a PhD student of Matt and Darius Armstrong James's, passed his PhD Viva back in 2019, and now is doing better than me, works at King's College London. He's quite a high up clinician now. And he did this amazing UK surveillance of azole resistant Aspergillus, and he saw that in clinical isolates, azole resistance was in 13% of isolates, and they were mostly from cystic fibrosis. And the TR34L90H combined resistance polymorphism was found in 27% of those azole resistance. So actually, it's quite high. And then Tom Sewell and Yu Zhang, I said this before when Caitlin attended my last talk, they literally went on this jolly across like southern England. Like they went all the way up to like Cambridge, down to Brighton and Bournemouth, Bath, and they just literally planned this trip over, where would be good to stop for a drink at the pub, and then they collected samples along the way. It was, it was a very good sampling method. So azole resistance in environmental samples was actually lower than in clinical samples. And then what was really interesting, remember when I said about the Burkhardt, we expected to see more azole resistance in the rural fields than in inner city London. Well, actually what we saw was that in rural fields, resistance was 1%, but in urban flower beds, where the flowers might have been imported from anywhere, we get a lot of our flowers from mainland Europe, but also the compost bags as well. So if you open a compost bag and test the compost, they're meant to be heat treated, but they're not heat treated sufficiently enough to kill off all Aspergillus fumigatus, sorry. So actually in urban flower beds, the rates of azole resistance was higher, which really surprised us. In the clinic, we're seeing increasing incidence of azole-resistant Aspergillus fumigatus. So we collaborate quite heavily with the Aspergillus group over in Manchester. They're also seeing an increase. So it's not just London, it's Manchester too. And then they're also seeing it in the Netherlands. We collaborate quite a lot with Paul Vervey and Jacques Meis and Ferry Hagen, and they're seeing an increase as well. So it's not isolated to just London or the UK. So Jen is currently stressing and trying to finish up her thesis. I've never known anyone plan to get married and hand in their thesis within 2 weeks of each other, but Jenny's going to do it. She decided she wanted to do like this environmental sample of the UK, but didn't necessarily want to go on the jolly on the road trip to do it. So she employed the citizen science approach, where she just got people involved and got them to sign up. And basically she just got a PCR seal, sent them out 2 PCR seals, and all they had to do was stick it down on their windowsill or somewhere sort of secure outside, and then send it in. So she did so just to collect air samples, she did 4 over 1 year. She also did a soil one as well, where she was inundated with aspergillus actually, and so she only did that once. She couldn't do it again for sanity. So this is a picture of the UK, and just gives you an idea of where she got samples from, all the way up into Scotland, Northern Ireland, Wales, basically the length of breadth of the UK. And you can see that there's perhaps a little bit of, azole fumigatus positive right in the middle, but generally speaking, there doesn't really seem to be much sort of spatial clustering in terms of whether you're azole positive or negative. And in terms of how many were resistant, the majority that were azole resistant were seen in the spring equinox and the autumn equinox, which was just surprising for us actually. We weren't quite expecting that. We were expecting a little bit more for autumn, when the leaves have kind of fallen, you get a little bit of rotting, a little bit of sort of like, decomposition. But actually it was more for the spring. But, in the UK, you have to remember that spring, the weather's a bit crap. And it's quite windy, it's quite wet, it's a little bit warmer, it gets a little bit muggy. So actually, this might favour aspergillus a little bit more. But you also have to take into account the fact that, she had a drop-off of participation over the year as well. So that might account for it as well. I think she has done statistical analysis on this to show that this is still significant. But I don't have those numbers to hand. But if you give her a follow on Twitter, she's very responsive to questions, even though she's writing up. So my main job is to use genomics to track azole resistant aspergillus through time and space. So we have just under 700 sequenced isolates. I am literally going into London straight after this because we've just had another 384 dumped on our laps. So that will take us up to pretty much 1,000, which is pretty cool. 400 clinical, 288 environmental, 2 other. I say other, they were kind of like experimentally passaged in a lab to kind of try and induce resistance. So I class those as other. Over 40 countries, 100 years. So 1919 to 2019, which is pretty cool. Yeah, and I think I said this last time. If anyone has anything from Canada or Russia, please send it in, because these are massive gaps at the moment. Yes, which we would like to fill. So what's really interesting is that we can track the azole resistance through time. So as you can see, in 1919, we are seeing this one here pop up. It's an American isolate, and it's wild type in CYP51A. So these are all CYP51A polymorphisms. And then as we go through time, we can start to see this left-hand side kind of start to fill out a little bit. We start to see some wild types fill out on the right-hand side, more wild types. We start to fill out over the world. And then come to 1998, we're starting to see the first drug resistance polymorphism. So we're seeing this one here filling out in the right-hand side, and it's this G54E mutation. It's somewhere in this Netherlands UK blob. We are quite heavy on the sampling in the UK and the Netherlands, to be honest. And then we start to see the TR34L98H's fill out in this left-hand side here. And again, they are in this sort of like this Western Europe blob. And then up to 2020, well, 2019 actually, when you can see that the majority of the TR34s are in the left-hand side. There are a few other polymorphisms in the right-hand side that aren't TR34. And there's then these kind of like these ones in the middle. There might be a TR34 here, which is actually an American isolate. But the majority kind of cluster in this left-hand side. So we denoted these 2 clades, we called them, and this is all down to Matt Fisher. I'll point this out. If you don't like it, complain to him. But he said clade A for clade azole and clade B for clade boring. So that's the reasoning behind that nomenclature. So in clade A, the azole resistance, you've got 227 that azole resistance, and wild type for CYP51A is 92. So it's 3-ish to 1 ratio. Whereas in clade B, azole resistance is 28, wild type is 338. So it's actually a 1 to 12 ratio. So clade A predominantly has the azole resistance. We've recently just had a paper accepted into Nature Microbiology. If any of you reviewed it, thank you so much. This has been this little project started 3 years ago as a little side project. I thought it would just keep me occupied, and 3 years later, it's actually become my life. We looked at just the genome diversity of Aspergillus within the UK, because the diversity within the UK is representative of Aspergillus diversity globally. And this was just over 200 isolates between 2005 and 2017. And the ratios are, you can still see that this clade A, clade B ratios are fairly, well-ish similar to what we see globally as well. But what we could see was that there was this clonal clade here, clade AA, because it sits within clade A. They're all TR34L98H, but they're incredibly clonal. And we see this globally as well in that, I'll talk about that in a bit actually. Oh, my phone's just made a funny noise. That scared me a little bit. So we were really interested in what this clone was. And interestingly, it's made up of 27 isolates, and they are 14 are clinical, 14 are environmental. So nicely split. They are all the same mating type. They're found from all over the British Isles and Republic of Ireland. So Dublin, Aberdeen, the south of Wales, London, and Brighton. And they're all TR34, which is very interesting. And they're all highly related. So basically they were separated by less than 300 SNPs, 244 to be exact. And I did a little t-test on this just to see whether we would expect to see that sort of randomly, or whether there is actually an association there. So the p-value was significant. So actually you wouldn't expect to see that sort of like level of SNPs between isolates. And anyone who's worked on Aspergillus and sequenced it knows that if you just take it out of the freezer, it will gain a few thousand SNPs. Like it is incredibly sort of diverse. The global pairwise average of SNPs between isolates is over 150,000. So these isolates are incredibly highly related despite their geographic dispersal. Yeah, that's just a little PCA showing that clinically, environmentally, they are quite close. So we think that this shows that a clinical patient could not have acquired this Aspergillus infection in any other way other than from the environment. We do have anecdotal evidence that people are potentially, particularly cystic fibrosis patients, they're coughing a lot, they could potentially expel Aspergillus in their coughs and things like that when they're coughing. But we think that this shows that patients are acquiring drug-resistant Aspergillus from the environment. This clade, as I mentioned, we do see it elsewhere in the world. We see it in Germany and Belgium and the Netherlands as well. So is it sort of like some sort of global superfit clone? And we also see it over in Thailand and Vietnam as well. And we know that the air currents kind of go sort of like west to east, and then they come over the southern hemisphere, and then they kind of come back again. So we are kind of expecting this clone to kind of sweep back around and start popping up in Australia, and also a US at some point in like the next few years. The UK and Thailand are separated by 500 SNPs. The Thai isolates kind of make their own little, like this little extra wing of the clonal clade. So what makes these isolates tick, we're not quite sure. I haven't quite had the time to figure that out yet. What makes these isolates tick? They're still similar, but they are slightly different. And then finally, just to wrap up, I did it, used this piece of software called TreeWAS by this guy called Xavier Didelot and a very talented, she's now a postdoc, Caitlin Collins. They wrote TreeWAS, which basically takes into account the phylogeny, the phylogenetic structure, when performing a GWAS, a genome-wide association. And you can basically also pop in a phenotype as well. So for instance, I just popped in a binary, is it resistant to itraconazole or is it not resistant to itraconazole? And as you'd expect, CYP51A L98H was the predominant, the most significant loci to come out, the SNP that encodes that. But then there was also some other interesting ones. There was just over 2,000 loci associated with itraconazole resistance. Some of them made sense to me, some of them didn't, and that's when we roped in the guys at Manchester, so Mike Bromley and Norman Van Ryn, just to kind of like look at this a little bit more. So if you just see this graph here, you can see that it's just like these peaks and troughs all over the place. It's not just like the odd loci. They all seem to be bundled together. So when you look at FST, which is kind of a measure of, it's a measure of fixation. It's kind of like a rough guesstimate to how loci are undergoing selection and things like that. When you line these up, you can see that these peaks and troughs line up with the FST. So you've got high FST values in chromosome 1. You've also got loci that are associated with itraconazole resistance. Chromosome 4, where CYP51A is located, you're also seeing these high regions of FST, and so on. It mirrors quite nicely. So there's these gene phenotype associations, and this FST is when comparing clades A and B, by the way. And the significant loci in these regions map to genes involved in secondary metabolism, including Fumitrem organ, which is why I need to get Mike involved, because he's really into this secondary metabolism stuff, and I have zero clue. So we got Norman to basically just do a really quick and dirty analysis. He just took 28 gene deletion mutants from the CoFun knockout collection and screen them for growth on media containing itraconazole. So CYP51A did not grow, which is good because it's been knocked out. We'd expect that. And it's also ABCA, and there has been a little bit of literature on this, so this is worth considering as well. But basically, just sort of going back, we need to test the flanking regions around these loci because there's clearly some linkage here going on, just to sort of like test those a little bit more. And this is only 28 loci out of over 2,000 loci. So and it's knocking out the whole gene as opposed to just taking out the specific loci. So it is a really quick and dirty approach, but it just kind of like was sort of a sanity check a little bit. So I'm just going to wrap up now because I'm realizing I'm getting to the end. So the public health threat posed by fungi is increasing, especially with the increase in COVID. I mean, we've seen what's happening in India. We've seen what's happening in terms of COVID-associated aspergillosis, so Kappa. We've got new emerging immunosuppressed patient case classes, especially those ones who were undergoing ibrutinib treatment, those who've had flu, those who've had COVID. New emerging multi-drug resistant fungal pathogens, C. auris. There's been a paper that came out in the last couple of months which suggests an environmental origin for C. auris. This is something that we've been sort of like speculating about for ages. And there's this paper, a really nice paper that's genome sequenced samples in coastal waters off the Andaman Islands. So this idea that it has a marine origin, which is something that Tom Chiller at the CDC and the mycology branch at the CDC have speculated for a long time. New patterns of antifungal resistance caused by dual use of antifungals. And we've seen in Aspergillus, this emergence of this potentially super fit clone, is high and which is resistant, it's capable of spreading. So there's this acute need to increase our focus and also the funding. I attended a meeting early in May, and apparently the number of projects for fungal research that have been successfully funded within the UK has decreased over the last 10 years quite dramatically. And the funding body actually said it's because they're not receiving applications, which I think, personally, I kind of scoffed at because I know that we submit applications, but it's a case of if you're not being seen, they're not gonna, if you don't ask, you don't get. So we really need to be kind of like pushing this. One thing that I haven't touched on is and is something that we do work on kind of like in the scenes is open access for surveillance systems. So this idea of just having this transmission specific analysis software. So when I was showing you the global and the UK Aspergillus, that was in a tool called MicroReact. And we are collaborating with the team that wrote MicroReact and also the CDC to kind of do, for first off, a Candida auris sort of surveillance software, which includes transmission analysis as well. And then hopefully we can expand that to other Candida species, because they're generally the easiest to work with, then crypto, then Aspergillus, because Aspergillus is a bit of a pain to work with. So this could be the base of establishing more appropriate policies and practices, which is desperately needed, especially in this COVID world. So just to wrap up briefly, I have to mention the funders because, you know, yeah, they've been good. They've been good to us. Imperial, the team here, Thomas and Amelie work heavily on Aspergillus, and obviously Darius as well. PHE, we work Public Health England. We work closely with Liz Johnson and Andy Borman there. So thank you very much for your attention.
Thank you very much for your talk. That was that's fantastic. Really interesting.
You're welcome.
What's NERC, by the way?
Natural Environment Research Council, which technically doesn't exist anymore because all the research councils in the UK just kind of got amalgamated into UKRI, which I'm not entirely sure what the RI stands for. But basically, they still kind of exist in theory, but not officially, if that makes sense. So, yeah. Yeah.
Right. I'm sure that there are going to be quite a few questions, so I'm going to get mine in quickly first. So we saw the same thing as you observed in the UK and Vietnam with the difference between resistance rates in rural versus urban areas. It was much like the highest place was the urban areas, as we had not hypothesized. So what's your thoughts? So we don't have the same thing with flower beds, so we can't blame it on flower beds in Vietnam. So is it an urban thing? You got any ideas?
No. Is the short answer. No. Because yeah, exactly. I mean, normally it's weird because when Jenny did the soil sampling, you know, sort of like citizen science thingy, I went around all the hospitals taking samples of all the flower pots outside the hospitals, and nothing grew, which was really odd. But saying like people's homes, stuff grew. So I don't know whether there's pollution sort of having an impact there, because traditionally, well, you know, where the hospitals are, if you think about Imperial, if you think about Paddington, St Mary's Hospital, it's highly polluted, whereas where people live tend to be in sort of like still urban but less polluted areas. So that's kind of like the only thing I can consider. It's kind of a balance between there's lots of people about, there's lots of sort of like activity from humans, but the pollution levels are slightly lower. I don't know. Yeah, it's a bit odd. I don't know. Yeah,
I've got.
It's an interesting one. We weren't expecting it.
We came up with ideas for what's happening in Vietnam, but they definitely don't apply to what's happening in the UK, and it's the same as you. So there must be another unifying reason.
Exactly, but and we just don't know what it is. Yeah, exactly. I haven't got a clue. Yeah.
Who wants to go first with a question?
Oliver?
You can just, I can't see everyone at once, so anyone.
Oliver's waving.
Yep, Oliver. I was waving. So flower beds. I don't know if you know Tom Rogers in Dublin.
Tom is lovely. Yeah.
I used to be one of his minions, and he was trying to convince me to send students to garden centres.
Yes.
A couple of years ago. And so the thought was he, knew mice and vervae, and they had the idea that it was tulip growers were one of the sources of azole resistance. Was that ever borne out? Because
so he carried on with that idea, and he had a postdoc called Katie Dunn, and they published. Yeah, and she's now gone into industry and is very happy. I think she's had a couple of kids as well, so she's extremely happy. She's flourishing. And they actually analysed tulip bulbs, and they published a paper on it showing that the tulip bulbs were kind of the harbingers of azole-resistant Aspergillus into the UK from Holland. And Jacques Mies was not happy. He was personally offended by it. But what was really interesting is then we we sort of like followed up with that, and we sort of, okay, well, you know, tulip bulbs, okay, but what about onions? They're still bulbs, right? So can we test onions? So we basically had a student go down to like the organic market versus Tesco, and just see the rates of azole resistance in organic onions compared to Tesco onions. And again, you can see the same sort of thing. And we also had someone test tea as well, tea from India/Burma, and just looking at the amounts of azole resistance in tea as well, and it's there in your tea bags. Perhaps not PG tips, but you know, perhaps like sort of the nicer stuff, the twinings. Yeah, it's amazing.
And is this quite a problem to sell? Because obviously farmers don't like the idea that they can't really use their favorite toys.
No, it's very hard to find a farmer who's happy for us to sample, because, you know, if we say, hey, we're trying to find this, you know, pathogen in your soil that could potentially kill people, they'll just be like, no. No, can't do that. And you can't exactly sort of like hop over the fence and just take it without their knowledge, because that's unethical as well. So sort of like the sign up rates are low, which is why the Burkhardt, the air trap was so good, because you could just put that on your land technically, but next to a field, and they can't say anything about it. But soil, different matter. Yeah.
Thank you.
People are funny about it. Yeah, thank you.
So we have questions. Maybe we go Tanya, Caitlin, then Julie.
So thank you very much, Jo, for a really interesting presentation.
You're welcome. Thank you.
I was really interested in your comment about the association of fungal disease with COVID patients, because we've really not seen it as that much of a problem. Admittedly, we haven't had anywhere near the number of cases you have. But I wondered if there were some specific associations. The things that come to mind clinically, I suppose, is a high incidence of underlying lung disease, for example. But are there any associations that you've picked up in relation to COVID and different fungal infections?
So I mean, I'm not a clinician. So when Darius like tells me about stuff, sometimes I'm like in one ear and out the other, because I have no idea. But it seems to be predominantly in those that have been mechanically ventilated.
Right. Okay.
And we went that brief. I don't know how much the coverage was over in Australia, but we did set up like these Nightingale hospitals, which were COVID specific, and they didn't last very long because we didn't have the staff to actually man them. But the one in London, there were incidences where Candida auris was actually getting stuck in the ventilators. And they Candida auris, it's very, you know, it's tricky to get rid of anyway. So we were also seeing Candida auris as well as Aspergillus in those that have been mechanically ventilated. But in terms of like other fungal diseases, we have had this outbreak, and I cannot for the life of me pronounce it. It's something clavata, something beginning with S, so Saffrichet or something like that, Saffrichet clavata. We've been seeing that in people who aren't necessarily mechanically ventilated, and this is at University College London Hospital, and that's been pretty devastating. Actually, that's been pretty horrible. So the clinician leading that is Neil Stone. He used to work for Johanna on crypto, and now he's sort of, well, I think he still wants to work on crypto, but COVID intervened. So it's kind of, I say the majority of times it's just those who have been mechanically ventilated. If there's any sort of, and if there's anything else, I don't know. I'm not that clever. I'm not sure. I'm not that sort of like on my clinical knowledge. You know, it'd be like Johanna and Darius would know that more than me. But yeah, for our point of view, for Aspergillus, for Kappa, it's just the mechanical ventilation. We've just had more of it because we haven't handled it well, have we, here? So
really
yeah, we're seeing loads of Kappa over in America as well. So
yeah. Okay, thank you.
Okay, Caitlin.
Thanks. I've got a technical question, and feel free to defer it. And I might be, so when I last saw your presentation, we're looking at the tree-wise analysis that you did, and we're looking at potentially doing it on Candida tropicalis, and I haven't progressed it, but from a limited reading, it's more for haploid organisms. Do you have any comments on its application, any challenges for diploid organisms?
No, I don't have any comments. I haven't tried it on a diploid organism. In theory, it should work fine because you're just going to have heterozygous and you're just going to have a mix of heterozygous and homozygous SNPs to deal with. So I think it's just a case of, yeah, I think that will. I think it would work, to be honest. I think probably the best thing to do would be to run it through something like gubbins or clonal frame, just to remove any sort of like regions of recombination as well, which might make it a little bit easier. And then run it through TREWAS. But in theory, it should work just fine, really. Yeah.
Thank you.
Give it a go. Give it a go. See what happens. If not, Xavier is very responsive to emails because he wants people to use it, right?
Okay.
And so he will do whatever he can to help that happen. Yeah, especially if it's something new and exciting. Yeah.
Thanks, Julie.
Thanks, Joe. That was a really nice presentation. I just wanted to ask you. So, it looks like the resistance that you're getting is not confined to the to the ergosterol biosynthetic pathway. Is that right?
Yeah, exactly. Yeah. And that's actually one of the reviewers' comments as well. And it's just a case of, okay, how do we show that? You know, how do we test that? So which is why we've got Mike Bromley on board, because that's kind of what he's going to run with for the next, you know, God knows how long, to kind of try and show that it's just so it's more complex than we realize, and it's not limited to just one pathway. Yeah, it's just like some sort of cross interaction.
Mention to you that we were at an ASM meeting last week, and Kylie Boyce, who works on Cryptococcus resistance, she works in Melbourne, I think at La Trobe. And she gave a really nice presentation demonstrating the same thing in Cryptococcus. So it'd be really valuable to look at that literature. Because,
exactly. And obviously with Cryptococcus, you have ploidy changes which contribute to the resistance. And that's something that we don't look at in Aspergillus, because we know there's drug resistance polymorphisms, you know, single amino acid and TR mutations in the promoter. And we just go, that's it. And actually, in terms of, you know, CCNVs and copy number variation or whole chromosome or duplications, we just don't look at it. So actually, that could be something, and we've completely missed it. And it was actually one of the reviewers' comments. So I've got my work cut out for like the next few months, to be honest. So definitely, yeah. What else we got? D.
I've got D.
Thanks, Joe. That was a really interesting talk. I was really interested in the phylogenetic tree that you showed, where you have so many azole resistance ones on one branch and very few on the other.
Yeah.
So, and I'm assuming that on the one with lots of them, that they aren't just from a single origin, that they've evolved independently. At least a lot of them have.
Well, that's next on my to-do list, to see whether TR34 L98H has evolved from a single origin or not. Yes, that's going to be quite a complex analysis to do. But now we're starting to see TR34 pop up in the other clades. Is that some sort of recombination, like a massive recombination event, and it's switched over, or is it, you know, a second spontaneous emergence? When we looked at it in terms of just Straf typing, just microsatellite typing, we could see that, well, we hypothesized that it just emerged on a single background, a single genetic background. But clade A and B are so different, we don't think that is the case anymore. But that I'm putting like a huge lump of salt with that, because I haven't tested it, but it's on my to-do list.
So do you think, if that's the case, that clade B will become highly resistant as well?
Exactly. So already, so clade B is the one that's got all the G54 mutations. It's got other sort of like weird, it's like just single amino acid changes that confer drug resistance. Whereas clade A tends to have the more complex polymorphisms that are also linked to a tandem repeat. So clade B is kind of ripe, really, for sort of like spinning out more drug resistance mutations. Or is it a case that we just haven't sampled enough?
Right.
Because the genetic diversity is huge. The panel that's waiting for me in London at the moment is full of isolates from Jen, and she sang the sequence, some of them, and just the polymorphism she's got are just so vast. It's incredible. So, you know, if you were to sort of like come back to me in a month, I'd be like, actually, do you know what? The pictures can change completely. So it's just constantly changing.
Right. Has anyone ever tried to do like experimental evolution on these strains just to see if you can evolve?
No, it's actually something that I keep putting in for funding, to do experimental evolution on a range of organisms, including Aspergillus, also Auris as well, and it just keeps getting rejected. It's just
too bad.
Yeah. So if you know of anyone who's got a spare pot of money, yeah.
Not here in Australia.
I'll keep trying though. I'll keep trying. Yeah.
Thank you.
You're welcome. Thank you.
Really interesting discussion as well. Does anyone else have a final question before we wrap up?
Yep. Can I ask
Please.
a quick question, slightly related to D's? I don't know how much this will make your life complicated, but going back to the phylogenetic tree,
yeah,
have those been mapped to the, you know, subspecies in the Aspergillus fumigatus complex?
Oh man. So yeah. So basically they're all mapped to AF293.
Okay.
Which is, in my opinion, a bit of a crappy reference. Because when you look at the sort of the amounts of insertions and deletions, you can see that there's, you know, either there's been some massive deletion events in Aspergillus in general, or AF293 is actually just a bit weird in itself and isn't representative of Aspergillus in general. So we were kind of waiting on CA10 being resequenced and RNA-seqed to have the gene annotations, and that was being done by Paul Bowyer. Still waiting on that. We've been waiting for a while. So basically, we've just got funding to do 4 PacBio Aspergillus sequences plus RNA-seq. So that is currently being done. Got a bit of a problem here in that because like some people voted to leave the EU, we can't get stuff shipped from the mainland to do our sequencing, which is a bit of a problem. It's all stuck in customs. Like, who knew that would happen, hey? Anyway, politics again. So we're doing 4 PacBio sequences to get some high quality reference genomes. Basically, we're going to do 2 clade A's and 2 clade B's, 1 environmental, 1 clinical, for each, in order to basically have clade specific references. Yeah, okay. And hopefully that should help resolve.
I realized I should have chosen my words better because you're a bioinformaticist and I'm more of a mycologist. So saying mapping them was, you know, not necessarily about sequencing. It was more about, have you identified to the subspecies level each of your isolates to see if some of the clustering
I see what you mean. So were they checked beforehand? Yes. So they're always Molditoft and whatever is required. As I said, I'm not that great in the lab. So we've got this lovely person called Sam Hemmings who does all of that, and prior to that it was Ali doing everything. And occasionally some slip through the net, so we do end up with some that we think are Aspergillus fumigatus and they turn out to be something like Neolypticus. But they're obvious when you put them in the phylogeny.
Yeah, but even within fumigatus, it's not a strict
it's not species. It is a cryptic species completely. Yeah,
so
exactly. So they are all
variation is related to that.
Exactly. So they are all checked for that to make sure that they're sensu stricto.
Just with you said you put a spore collector out at Rothamsted.
Yeah.
Did you speak to them about looking at some of their really old soil samples?
So that's obviously Bart Fry.
Yeah.
And then
I know Bart a little bit.
Yeah, Bart is no longer at Rothamsted. He's somewhere else. I can't remember where off the top of my head. basically Rothamsted cut their staff last year dramatically, so he's had to go. So yes, it was one of the things that was kind of a deliverable on the NERC grant, which finished last year, and then we got the Wellcome Trust grant to look at clinical Aspergillus. But given his institute move, that hasn't quite panned out. But it is, yeah, we know that they've got extensive air and soil samples. So yeah, it's on.
If you need another contact there, I can probably put you in touch.
Good, cool. Thank you. Yeah, because we've lost all insiders at Rothamsted now.
Okay, well, I can get you hooked up, I guess.
Yes, nice, awesome.
Cool. Thank you, Joe. That was a great talk. Oh, Caitlin, have you? No, just a great discussion. I thought I should snuck an extra question in. We are out of time.
Thanks, Caitlin.
So
that's fine.
Thanks a lot for making the time. It was really.
That's all right. Go home, have dinner.
Thanks very much, Joe.
Thank you for joining us. I hope you've enjoyed today's talk from the Fungisphere webinar series, and we look forward to seeing you next time.